So, good morning or good afternoon, depending on where you happen to be in the world, and welcome to the second in a series of Beyond Borders webinars, run a collaboration between the Industrial Biotechnology Innovation Centre, or IBioIC, and SINERGY. The teams from both organisations first met at World Biomarkets last summer in the Netherlands, and in conversation we recognised quite quickly there was lots of complementary activity across both our networks. And so we hit on this idea of running some awareness raising webinars in the hope of stimulating collaboration between our networks. My name is Kim Cameron. I'm one of the senior impact managers at IBioIC. And from the Singapore side, we have Rachel Lowe, industry development lead, joining us. It's just a little bit of housekeeping before we begin. The session's being recorded and we'll post a copy of the recording to our website shortly after the session concludes. If you want to put your questions in the Q&A box, we've got a short Q&A section after each of the presentations that we have coming, and we'll do our best to answer them there. We do only have an hour this morning, so if we're not able to answer all the questions in the live session, we can pick up with you offline. And then you'll notice there's a chat function as well, so that's for you to communicate with any of the other attendees at the webinar, and also to let us know if you're having any technical issues at all. So we'll be together for an hour. Shortly, I'll give you an introduction to IBioIC and then hand over to Rachel, who will tell you a bit more about SINERGY. And then we'll have a series of pitches from companies and academic groups, followed by Q&A. So it'll be a 5-minute pitch with then 3 minutes of Q&A. And then we'll just finish off with a bit of information about next steps. So some background to IBioIC. We're based in Scotland, but work internationally. We're a hub between industry, academia, and government to help accelerate and de-risk innovation across the bioeconomy. We run events like today's, and we also have our conference coming up, which I'll tell you a bit more about in a moment. We provide funding for industry-led collaborations with Scottish academics, and we have a scale-up facility to help people develop their bioprocess. And we also help deliver a programme of skills and training from HND through to PhD and some CPD as well. IBioIC was set up in 2014 to support Scotland's national plan for industrial biotechnology, and this provided a roadmap through to 2025. So we're currently working with key stakeholders to refresh the plan, and we're very pleased to say that we believe we exceeded the targets set out in the initial iteration of the plan, and excited to see what comes next for IB in Scotland. So IBioIC has a network of over 420 industry members from across the world, so Scottish, UK, and international, all shapes and sizes and all flavours of biotechnology. And we're also well plugged into Scotland's excellent academic base. There's a snapshot here on screen of some of the research expertise across the country. So I promised you a plug for the conference. Here it is. Our annual conference, the UK's largest, runs in Glasgow on the 10th and 11th of March, so not far away now. It's perhaps a little bit far for those of you in Singapore to come along if you don't happen to be in the area. But if you are in the area, we'd be delighted to see you. There's a QR code on screen for you to register. You can find out more on our website as well. So please do join our network. It's free to join. And again, there's a QR code on screen, and you can also register and join the network via our website. So with that, I'll hand over to Rachel, who's going to tell you a bit about SINERGY.
Thank you. Thank you, Kim. Let me see if I can control the... Yes. So I think I could control. So good morning to the Scottish attendees, and also good afternoon or evening to the Singapore attendees. Like to give our appreciation to our counterpart in Scotland, IBioIC, for the dedication. And also this is the second edition of our Beyond Border series. So I'm Rachel. I'm from the Singapore Consortium for Synthetic Biology, and we are here to support the synthetic biology innovation in Singapore towards a global bioeconomy hub. We are a National Technology Consortium. We are there to serve and consolidate and harness the synergy in Singapore's capability in synthetic biology across the industry sector to create a vibrant and globally connected bio-based economy. The first R&D synthetic biology program in Singapore was set out in 2015, and it's called NUS SynCity. But as the industry traction and interest increased, Synergy was launched in 2017 to facilitate the academia industry collaboration. We also work very closely with the enabler, such as the Tech Transfer Office and also the government agency. A snapshot of our academic and also industry member. We have our member from the majority, all the major research institute. These include the National University of Singapore, Nanyang Technological University of Singapore, Singapore Institute of Technology, A-Star, and also the different polytechnics. We currently have 37 industry members, ranging from startup, SME, and also NMC. We aim to create platform to catalyze industry academia collaboration. We actively participate in international conferences, and we also host exclusive or open workshop for our members. And we also host synergy seminar and also webinar as such to facilitate and promote cross-border collaboration. We are a very lean team, but we hope to serve the nation. And if you have any question, feel free to drop us an email or follow us on LinkedIn. Thank you.
Thanks, Rachel. Thanks for the intro. Yeah, we're going to hand over now to the first of our speakers, Philip Shelton, Chief Operations Officer at IBio. I think you're muted, Philip. Are you able to come online?
No, figured it out there. I just lost my mute and video button there. But yeah, thanks for the introduction, Kim. So I'm Philip Shelton, Chief Operations Officer of IBio. And what we do at IBio is we make peat-free growing media, also called potting soil, from locally available raw materials. And we support commercial growers. So think glass houses, the people who grow lettuce for your salads, trees for our forests, and of course, petunias for our gardens. Those are our clients, and they require a peat-free solution because all across Europe right now, there are restrictions and bans on using peat, which is was the most commonly used raw material. But now, with restrictions and bans coming in, preventing its use in horticulture, there are now a need for peat-free alternatives. And the reason why there are now restrictions and bans on peat is that peat emits a huge amount of carbon because peatlands, which are swamps essentially, are effective carbon sinks. They store up to, I believe, 4 times as much carbon as a single hectare of forest. So when you dig that peat up, releases all that carbon. So it's a major issue when we're using it to grow plants because then the carbon emissions are released at that point. So that's why peat-free alternatives are needed. And for us, we're manufacturing at TRL 7, so we're manufacturing at pilot scale. We have patent pending compositions and production processes. And the interesting bit about our production process is that it allows us to use a wider range of raw materials than what you would typically see from our competitors and others in the sector. So we're able to take a local first approach and a circular mindset when we design our products. And we have already done trials with professional growers who have paid for our product, and they've seen better water retention, faster rooting, improved seed germination. So, it's not just a theoretical exercise. These are benefits that growers are seeing firsthand. And again, another thing to bear in mind is that even if there were no bans on peat use to grow the plants that we need, there have been supply chain issues with peat. With just last year, 2025, Europe only harvested about 50% the normal peat volume. So it is a food security issue at this point. And I was just speaking with a Spanish grower who couldn't secure the supplies of peat that they normally would need and require. So that's why they're considering going peat free, because they just can't get enough peat. And that's a story that you hear time and time again in our sector about why growers need options like ours. So ours has been validated across a variety of crop types. So we've done vegetables and propagation blocks like you see there. And in fact, I have one right here. This is a pepper, by the way. So fun there, just germinated. And we've done it for forestry in mini plugs and, of course, in pots for ornamental plants and herbs. And our trials have shown a 5% improved germination and up to 15% improved plant shelf life. And for one of our clients, who is a major supermarket supplier, that's over 6 million in potential revenue gain just from those two improvements alone. And the feedback speaks for itself. They saw fastest rooting and best water retention. And ultimately, as a company, we are happy to work and collaborate with other interested parties. And if you're interested in trialing our product, always happy to provide that. And we're also raising an investment round to take ourselves to TRL 8 and 9 in order to produce more of the product that growers are requesting of us. So thank you for this opportunity to pitch, and I will pass it back to you, Kim.
Thanks very much, Philip. Any questions for Philip? Oh. Eve, looks like you've raised your hand. If you want to.
Hi, Philip. Yeah, that looks absolutely amazing, especially that it is even more improved benefits compared to peat. So, I've got two questions really. Number one, why do you see those benefits compared to peat? What is special about the material, and when you do commercialize and reach TRL 9? What will your price point look like compared to the price of peat today?
Oh, that's a great set of questions. So to answer the first one about what makes our material special, there is a second part to our process that I didn't touch, and that's the biotechnological part. So it's not just that you mix a bunch of materials in a pot and then you're done. For us, we also employ biotechnological approaches. That's more my co-founder's area of expertise. But in essence, we select materials very carefully to harbor a specific type of microbiome, which is beneficial for plants. So that's the long story short of it. But there's also some other things that we do that are quite interesting. And to answer your second question about the price point compared to peat, I'm glad you asked, because it is one that growers commonly ask us. And for us, once we're fully scaled up, we are intending to hit price parity with peat-free options and peat. And here, it's important to note that right now within Europe, peat prices are lower than peat-free. However, the projection is that in the long term, that price has only been going up over time. So the past decade, peat was cheaper 10 years ago than it is now because of the supply chain issues I referenced before. So even if, we don't do anything to decrease the price ourselves, the peat price is just going up and up and up. So hence why there's a need for peat-free alternatives, other than the good environmental reasons and the regulatory ones.
Okay, thank you.
Oh, no problem.
Thanks, Philip. As I mentioned earlier, we're a bit tight for time, so if you don't mind, I'll move on to our next speaker, but thank you.
No problem.
So our next speaker is Siren Lim from the Nanyang Technological University in Singapore.
Okay, hello. Hi, happy to be here, and I'm going to present some of the work we've done in the lab. Okay, trying to advance the slide. And yeah, so our lab is basically engineering biological molecules from proteins to microbes. And the applications that we try to use them for is for bio upcycling of plastics, production of materials, in this case, cellulose, for health application that includes skincare as well as wound healing. And in the last 5 years, we have been working on this plastic bioupcycling project where we design new enzymes to degrade PET plastics and feed the monomers of these plastic hydrolysates to microbes. In one of them, we try to produce cellulose, and then the other ones, we try to produce fatty acids. And on the other part here, to kind of like make it circular, we had cyanobacteria, where it takes the carbon dioxide and then produce the enzyme that will then break down the plastic. And through these exercises, we have then developed a high throughput screening technology we call SPEED, screening platform for enzyme enhanced degradation. And that basically allows us to identify best performing enzymes, and we can screen about 1,000 variants in 2 days. So that speed basically is important for our enzyme engineering efforts. And hopefully that will help in terms of clasping the loop in terms on plastic and conversion of that plastic into some other molecule. So building on this capability, we have recently received funds to collaborate with fantastic groups in the UK. I have my co-PI here, Joanna Sadler, on the call, and she will be happy to also share some of the activities that we will have. So basically, in this particular effort, we will be degrading different types of plastics beyond PET, and then engineer the microbes. These are the 3 microbes that we will be engineering to produce these 3 specialty chemicals. And this is of interest to Singapore, as well as in the UK. So this is the goal of the joint project. And of course, along the line, while we are trying to develop engineering biology tools, we realize that we need to ensure optimization as well as production. And beyond that, the need for standards and measurements that we will be sharing between multiple groups. So we have 4 different universities and 4 different, more than 8 research groups basically are involved in this project. And lastly, here we also have automation, lab automation that will be developed or also in here. So if you are interested in looking into or joining us in our quest in this new project called CircBioNet, which is circular bioeconomy through engineering biology for specialty chemical network, we have a upcoming public day, and I'm probably going to invite Joe to give a little bit overview of the public day. Joe,
Thank
please.
you very much, Siren. Thanks for the introduction. So this is an event on the 26th of February, which is going to be at King's Buildings at the University of Edinburgh. And this is an open seminar with short talks from all for most of the CircBioNet PIs, going to give a brief overview of their lab's research and plenty of networking opportunities within that. So it will be attended by a lot of CircBioNet representatives and as well as a lot of engineering biology community from across Edinburgh University and also further afield within Scotland. So this is an open seminar. If you're interested and you would like to come along, please do sign up. There is a joining link at that QR code. And also send us an email if you've got any other questions about it. But hopefully to see you, we'd love to see some of you there. Thank you.
Thanks.
Yeah, thank you, Joe. And that's all we have for our project.
Thanks, Siren. Thanks, Lim. Does anybody have any questions for either Siren or Joe? Okay, we'll move on to our. Oh, sorry, Huiling, do you have a question?
Yes, hi Sirin. I'm just curious about your project. I'm curious, where will you get all these waste plastic feedstocks, and at what purity must they be at for you to perform your metabolic pathways?
So the one that we have been working on for the PET, we have just used water bottles at the moment. And that will just coming from a standard waste collection facilities. And in the future, we can work with waste management systems in Singapore, because what we understood is that all these plastic bottles, especially for the single-use drink bottles, those are shredded, and then we can basically use the shredded ones into the process. And one of the nice things that I didn't mention is that the hydrolysate that we recover from the enzymatic degradation does not need to be further purified. The microbe can actually selectively pick up the molecules that they want to use for the conversion. So our lab has done it for making cellulose, and one of the monomers has been used as a carbon source. But Joanna's lab has actually managed to do conversion into paracetamol, for example, and among other things. Yeah.
Awesome. Thank you so much.
Thank you.
Thank you. It's really great to see a current example of a Scotland-Singapore collaboration. So yeah, really great. I'll hand over now to Alex Speakman from EVA Biosystems for our next presentation.
Hi, everyone. So I'm Alex. I'm the founder of EVA Biosystems. Now, as I'm sure we're all aware, plastic pollution is a huge global issue. It's estimated that over 14 million tons of plastic enters the ocean every year, and none of it is going to degrade anytime soon. But there is a reason how we got here. Plastic is amazing. It's strong, it's cheap, it is everywhere in our modern lives. But while we're doing our best to develop new alternatives to plastics like bioplastics, they are simply too expensive and in too short supply to truly replace plastic. Now, as a bioengineer, I've seen synthetic biology applied to a whole host of other global issues, and I know that this is no different, and I know that we can do better. So this is what my company, EVA Biosystems, is trying to achieve. We are developing selectively degradable plastics. We aim to maintain the benefits that made plastic so widespread in the first place, while tackling plastic pollution at a meaningful scale. So our technology comes in the form of engineered microbes to be embedded into the plastics themselves. These microbes can survive going through the manufacturing process, lie dormant through usage, and then become activated by a specific environmental stimulus to then produce a plastic degradation enzyme. Now, by working as an additive, we want to work with the low cost and abundant plastics that are already used by industry, meaning we can avoid supply chain disruption, be attractive to large-scale manufacturers, and make the biggest impact possible. So the ultimate goal for our technology is to produce a plastic packaging that can sense if it's entered seawater or landfill and then degrade itself in response. Could I have the next slide, please, Sophie? So just to illustrate the type of work we've been doing, so we've been making our own 3D printed filaments with our microbes embedded inside. These are designed to produce a fluorescently tagged degradation enzyme in response to a seawater level of salt. So we can print out our parts, put them in salty or non-salty, and as you can see, they can detect that change and respond to it enzymatically. And while we're still very early stage, we have been able to achieve degradation of our composite 3D printed parts, so quite proud of what we've been able to do. So as a business, this will work on a B2B business model where we sell biological material as a paste or powdered directly to plastic packaging manufacturers or to resin petrochemical companies further upstream. This means that we can focus on the biological side of things, such as bioreactor scale up, strain optimization, and not have to build a plastics company from the ground up. But we also have 2 patents filed so far, so we can also look at licensing as a way of engaging with the market. Speaking of the market, it's large. We're currently targeting PET as our polymer of choice, and this is worth roughly $35 billion globally. But as this is an engineered platform, this is a platform technology, we want to customize it with different enzymes to different polymers, different degradation activation triggers. This could be even larger. So we are a small team of 4. We are all PhDs from the University of Edinburgh, ranging from synthetic biology to polymer chemistry. So we're scrappy, we're lean, but actually we punch well above our weight in terms of our capabilities. Could I have the next slide, please, Sophie? So our current work is we are working on a bio-based manufacturing call where we are looking at taking our work with 3D printing and progressing to the injection molding of PET. So what we wanted to do is take this work and demonstrate it with a more commercially relevant plastic and in a more industrially relevant setting. So no one makes things with 3D printing. We want to show that more industrial use case scenario. So this is being done in collaboration with National Manufacturing Institute Scotland, who are part of the University of Strathclyde. So we're based at King's Buildings in Edinburgh. We can do the bio work here, and then we can go over to Enmith to do the injection molding work. So that's quite a nice division of labour. But this is all made possible by a recent investment from Coca-Cola Euro-Pacific Partners Ventures, and this project will conclude in this coming October. So as fundraising never ends, we're already considering what our next round is going to be. So we're looking at around 2 million to 3 million to continue R&D after the project ends, and also look at initial pilot projects and scale-up activities. So my ask to you today would be to help us make connections and expand out into the Asia Pacific region, and Singapore would be a perfect jumping off point for that. So we're looking for potential collaborators and pilot project partners that can help us demonstrate the use of the technology. But in particular, we're really keen to make connections with petrochemical and resin suppliers. So we are working with Coca-Cola as a packaging manufacturer, but really we want to make connections further up the supply chain to show how our technology can be applied to different customers and work out where the true value proposition is. And of course, with the fundraising, if you are investors yourselves or know people who this might be the type of thing that they're interested in, I'd welcome anyone to kind of send us their way. So thank you very much for your attention, and please feel free to get in touch. Thanks.
Thanks, Alex. Does anyone have any questions for Alex? Oh, okay, well, we'll move on to our next speaker in that case. Erica at Casville Labs, I'll hand over to you now.
Yes, thank you very much, Kim. Hi, everyone. Pleasure to be here today. My name is Erica Raivala. I'm the CXO at Casville Labs, and with me today to help answer your questions is our head bioinformatician, Teo Huttunen. Casville Labs is a software and bioinformatics company that operates in the health and pharmaceutical space. Our mission is to develop elegant and efficient software that always has the end user in the center. Our vision is to become the de facto infrastructure that powers scientific breakthroughs. Our team is very versatile, and we mainly hire T-shaped individuals who are very good at what they do, but also motivated and capable of growing to new roles. Currently, we have engineers, behavioral scientists, ethnographers, experts in bioinformatics like Theo here, genomics, psychology like myself, and business and commercial strategists. Let me go to the next slide. Hold on, please. Perfect. So Casville Discovery is the tool that I'm speaking about today. It's our unified drug discovery platform that accelerates pharmaceutical R&D with artificial intelligence. We have over 40 datasets, almost 40,000 phenotypes, and multi-omics profiles for over 60,000 targets. We deal with all kinds of data and are always working on widening our database selection. We facilitate complex analyses, offer high-quality visualizations like cellular localization and 3D modeling, and support customized workflows, all with an AI copilot for the biggest scientific impact. One of our main focus areas is efficient and transparent collaboration, and that's why we have created various features and spaces for researchers to communicate and collaborate around data without things falling through the cracks. And as you can see, we already have quite a lot of traction going on from our ongoing pilots. Today, we're looking for co-development partnerships, pilot partners, data partnerships, industry experts in drug discovery, multi-omics, biological research, and the clinical side as well, to provide us and to develop new use cases to help us drive focus, clarity, and momentum at the forefront of data-driven drug discovery. That's all from me today. Me and Theo will be very happy to answer any of the questions you might have. Otherwise, thank you very much for your attention.
Thank you, Erica. Anyone have any questions for Erica before we move on? No? Oh, Liz, you've raised your hand.
It was more a suggestion, maybe a point of collaboration. So we have a sibling innovation centre called the Data Lab in Edinburgh, and they're very well connected around data, artificial intelligence. We also have a really kind of world-leading group called the Bayes Centre in Edinburgh. So it might well be worth you kind of reaching out, connecting with them to see whether or not there's some points of collaboration or synergy with some of the expertise that we have in Edinburgh around AI and data.
Absolutely.
So yeah, I think there might be some interest there. So we can make those links for you, Erica.
Perfect. Thank you very much.
That's a good
We'll do
point.
that.
Okay, thank you. I'll hand over now to Eve Wellman from Ripsell for the next presentation.
Okay, hi. My name's Eve Wildman, and I am the CEO and CTO of Ripsell. So we are based in Scotland, in Aberdeenshire. So this is where in the heart of the whiskey country. So this is our team here, and this is a little bit about our journey so far. Similar to Alex, we've had some government funding and from the Innovate UK recent Bio Launchpad funding as well for Scotland-based innovations. We have run a pilot project with local whisky distilleries, and we have secured around 500 million litres of biomass feedstock through recent heads of terms that we've signed. We have worked with IBioIC last year for a feasibility study in collaboration with the University of Aberdeen. We are currently on an investment drive for £3 million to aid our scale up, and we are doing customer testing with a lot of partners at the moment, Lush Cosmetics, Associated British Foods, and several other cosmetics companies. And we're using the Innovate UK at the moment, that funding to scale up on our first flagship site in Speyside here. So this is it's probably one of the world's largest biomass refinery sites for distillery waste in the world. It takes the waste from 16 local distilleries, it pipes it through gravity down into the valley, and then they convert that into renewable energy. So we're working with them to try and add more and increase their resource efficiency and extract more valuable chemicals from that material before it goes to energy generation. We have a PCT patent pending in Europe and US territories, and we have an independent life cycle analysis report. So we convert whiskey distillery biomass into high value biochemicals. So at the moment we're targeting bioacetic and biolactic acid, which can enter multiple markets, but we're also doing a project at the moment that's looking at phenols, prebiotics, to try and expand this portfolio. We work with partners that also then take some of our upstream waste and convert that into biogas, so natural methane production through anaerobic digestion. And we also produce large amounts of clean water for our partner to use on site in cooling and cleaning activities in their infrastructure. So just before I finish here, I thought that this call and this relationship with Singapore is extremely useful. I think for Scotland, I was at an event recently where they were talking a lot about Jurong Island, where you have a sustainable focus on sustainable green chemicals, and they've actually built pipelines to local docks to export them out, and there's a huge amount of investment gone into there. So I really would think that it would be great to strengthen ties with Jurong Island and get some contacts there for people who'd be interested in working with us and our biochemicals. Also, I was hoping to look for strategic partnerships. If anyone on here has any information about anyone who would be interested in strategic partnerships for market entry into Southeast Asia, with industry stakeholders who are interested in cosmetics. So our product is a alpha hydroxy acid. It's biolactic acid, and it's been tested up to cosmetic and food grade. So we are looking for customers in that area. So yeah, if anybody has any ideas on collaboration, I'll pop my email in the chat. And yeah, thank you for your time. That's me done.
Thanks, Eve. I just want, before we move on, I wonder if anyone has any questions for Eve. Hui Ling.
Hi, Eve. I'm curious when you just wanted to understand more when you said you want to find collaborators on Jurong Island. Could you share a bit more details of what kind of collaboration you're looking for? I think that would help me as an ecosystem partner in Singapore to also find the relevant people.
Okay, yeah, so really, if there was anyone there with contacts into who are making sustainable chemicals and raw materials for the cosmetics industry.
Okay,
Thank you.
understand. So it's more on the formulation side.
Yes.
Okay, thank you.
Thank you, Lee.
Thank you. I'll hand over now to Addison Wong from Micrometer for the next presentation.
Thank you, Kim. Hello, everyone. Thank you for joining us. I'm Addison, and with me, Jean, who is in the panel. We are from Team Micrometer, and we are building the next generation rapid and reliable diagnostics for the pet care industry. This journey started from home. I have a toy poodle, Jolly. He's 6 years old this year, but when he was 3, he suffered from skin infection. And despite multiple visits to the vet, there was no clear diagnosis and no relief. Watching Jolly suffers without knowing what was wrong was difficult, and that's a pain that many pet owners can resonate with. Pets are becoming family members, and many families treat their fur babies' well-being with the same seriousness, care, and love that they do for their loved ones. Sorry, can I have the next slide, please? Skin infection is one of the most common and persistent problems in the pet care, and about 4 in 5 pets face recurring skin diseases. The cause is often unclear. How can we distinguish a bacterial and a fungal infection or even allergies? For a vet, it's not always straightforward. We cannot be giving antibiotics to an allergy. We cannot be giving steroids to microbial infection because both would make matters worse. Among skin diseases, ringworm is a highly contagious fungal infection that requires antifungal treatment, but is often missed because its symptoms overlaps with many other skin conditions. For current diagnosis and treatment, it can take more than 1 week, from fur plugs to waiting for microbes to grow on a petri dish. During this wait, the spores would have spread through the living environment, increasing the risk of future reinfection and turning a single problem into many months of frustration. Our goal at Micrometer is to change this with a rapid point-of-care test that allows diagnosis and treatment from day one. And let me show you how our product works. First, we will collect a simple skin swab and then dip it in our test solution containing a customized DNA probe. We then add a trigger solution. From swab to results, all in just 30 minutes, we'll witness a red to purple color change. The purple intensity will indicate a higher microbial load. These results can be captured with our proprietary smartphone app, and with machine learning algorithm, we are able to give a result that is quantitative. So it goes beyond a simple on-off quantitative test, just like a lateral flow, let's say. But beyond that, it allows vets to track infection level over time and verify whether a treatment is indeed working. These in turn translate to improved clinical decision-making, better aftercare for pet, and a peace of mind for pet owners. Our technology has been validated in an adjacent water testing industry, and we are filing patents in Singapore and other countries in Asia. So the product itself can expand beyond the current fungal test and includes a multi-pathogen panel or other microbial infections such as virus and bacteria as well. So this is the team that we are bringing the product to life. Jean leads the product development and development of sales strategy as the CEO of the company. I serve on board as a technical advisor. I'm based in the Singapore Institute of Technology, and together we bring over 20 years of experience in biosensor and infectious disease management and control. We are supported by our business advisor and commercial champion, Alvin and Debashi, who brings experiences in medtech productization and commercial sales in launching it to Asia Pacific market. We are supported by our vet partners who are senior clinicians in the Singapore pet care industry, as well as the regulatory services and agency. So we hope that you could lend us your support and journey with us as we build a We-First society, not just for human beings, but also for our pet babies who stays silently by our bedside watching us go to bed at night. We are open to meeting anyone for perhaps collaboration for product trials and testing, as well as seeking investment in the future. So thank you very much for this opportunity.
Thank you, Addison. Does anyone have any questions for Addison? Eve.
Hi, Addison. Thank you for that. My dog passed and he suffered with terrible skin infections right till the end. So it's obviously what you're doing is really, really important. As a past academic who's moved into this commercial space, I was interested to see that obviously you have IP protection, but you also have a publication under review. So have you got any advice for how you can continue to publish in the academic space, but also protect your IP from a commercial perspective?
Our current strategy is to file for the patent first, and for publication, once the patent application has been applied, then we will proceed with the publication. However, in cases whereby there are some journals that allows the work to be published without going into very detailed science of it. So we have actually come across a few biosensor journal that actually allow so. So these are possible avenues.
Okay, thank you.
Thanks, Addison. So we'll move on to our last presentation now. Ioannis Stasinopoulos.
I think Liz have a question.
Oh, sorry, Liz.
Thanks.
Sorry.
Hi, Addison. Nice to see you again.
Hi,
It's been
Liz. Nice to see you
ages.
again.
It's been ages. I just wondered, do you have any specific needs to support the development of your product that we could help you with? We have a lot of veterinary research and a lot around infectious diseases in Scotland. So, you maybe you don't need to say now, but if you do have specific things that you need to do to bring your product forward that you can't source in Singapore, then please do reach out and we can make some specific connections for you. So
Thank you, Liz. I will definitely reach out to you. We only have the local vet as our partner, but we hope to actually have more vets on board that will give us the necessary insights and input to develop product that is of a real use case.
Yeah. No, we're obviously a nation of dog lovers. I've got one myself, so we really have lots of volunteers. But thank you. It's really good to see these kinds of applications coming through, especially around really accelerating diagnosis and treatment for animals. So yeah, really like it. Thank you.
Thank you, Liz.
Thanks, Liz. Thanks, Addison. So yes, I'll hand over now to Ioannis Stasinopoulos, the CEO and co-founder of Prozimi Biolabs.
Thank you. Thank you very much, Kim. Thank you for the invitation. I'm trying to find how I'm going to turn on the camera because it just disappeared before I managed to turn it on. The camera button.
Down on the bottom left for me.
Yeah, it was the same for me, but I turned on the microphone and the camera button disappeared after probably I got the control of the. Let me double check here. Hello, everybody. Thank you very much, Kim, for the invitation, and apologies for the small technical issue. So I'm CEO co-founder of Presume Biolabs, and I'm going to try to take you on a journey on how synthetic biology and engineering biology can actually offer solutions to the food industry as well. We're Presume Biolabs. We're a biotech food tech company that we're based in Edinburgh, a little bit outside of Edinburgh at Roslin Innovation Centre. And to give you a point of reference, is the same campus that they cloned Dolly the sheep. And we are a biotech food tech company that we develop an enzyme technology that aims to introduce a new sector in the food industry, the gluten-safe sector, where gluten is still present in the product, but safe to be consumed by everybody. Gluten can be found in a number of different staple foods that we eat on a daily basis, such as bread, pizza, pasta, beer, and all these products, they are just like have a really kind of like good impact in the society, and we all eat them on a daily basis. Now, gluten has two sides: the good side that gives the nice structural properties to these products, they like the nice texture of the bread, and the bad side, where it's harmful for a number of people, and is actually around 8% of the world's population that suffer from gluten-related disorders. Now, the industry are trying to avoid gluten by using gluten-free flours to make all these products that I mentioned earlier, like rice flour, tapioca starch, potato starch, quinoa. And they try to mimic the regular bread, regular pizza, pasta, without just like, by failing actually to do that in terms of like the texture and the flavor of these products. That's why the vast majority of the gluten-free consumers out there, which is approximately 16% of the world's population, they're not satisfied by the texture, the flavor of these products, and the high price of these products. On the other hand, we have bread producers as well that they try to make better gluten-free formulations day by day, but for them it's also complex to do that because they use a number of different gluten-free flours, and they try to find formulations that they mimic regular bread, for example, without managing to bring back the texture, the nice texture of the bread. And it's also quite expensive because they buy these flours at least 4 times more expensive compared to regular wheat flour. Now, our solution, as you can see here, and I cannot change the slide.
Oh,
Thank you. Our solution, as you can see here, is that we can use gluten-degrading enzymes, and we can add them in the bread formulation as another ingredient at the stage of dough preparation. And in order to degrade and deactivate, if you want, neutralize the harmful parts of gluten, as you can see here, and keep the nice structural elements of it that gives a nice texture to the bread. So by doing that, we're offering to the bread producers a value proposition where they can go back to the regular formulations, use wheat flour instead of all the other gluten-free flours that they use at the moment, reduce the production cost, and simplify the process. And at the end of the day, they can give back to their consumers what the gluten-free, especially consumers, what they kind of like desire the most, a nice gluten-safe, as we would like to market it, bread. At the moment, and as you can see at the next slide, we have, thank you, we have developed a pipeline for screening our enzymes. And we're at the stage that we try to express them in model organisms in order to be able to scale up our enzyme production and start just applying it in the small pilots of bread making formulations. In order to do that, of course, we need to kind of like get regulatory approval from EFSA, the European Food Safety Authority. And of course, we're aiming to reach the market by 2029. In order to do that, of course, we're looking for 2 million investment. And of course, we're looking for partners/collaborators that they can help us with the scale up, with the commercial pilots, with the bread making pilots, and of course, for the regulatory approval. And at the next slide, we have a couple of QR codes that you can follow our website and our LinkedIn page. And let's make all together a gluten-safe world full of flavor. Thank you very much. And I'm open to any questions and any clarifications. And again, apologies for the technical issues here.
Thanks, Ioannis. Any questions? Rachel.
Hi, Rachel.
Hi. I'm just curious in terms of getting collaboration in Singapore, do you have specific collaboration in mind that you want to seek out in Singapore, or are you looking at more within the UK space at the moment?
I'm actually already in touch with and I don't know if it's just like government agency or just like more kind of a private company regarding the regulations, because I know in Singapore you have the SFA, the Regulatory Approval Authority. So I'm already in touch with them. I'm going to have a meeting next week. In terms of like the regulations, in terms of like fermentation, we haven't explored that, or in terms of like applying our ingredients in the bread making process, but we're more than happy to explore that as well. But yeah, we have already started reaching out in terms of like, because Singapore could be a really good first sort of say for us in terms of like the Asian market for later on.
Thank you.
Thank you.
Thanks, Ennis. Thanks, Rachel. Any more questions? So just a little bit about next steps. First of all, an apology. I'm aware that a number of attendees have had problems joining the session this morning. We have been recording the conversation, and we will share the recording in an email follow-up so that people can make contact, and it ticks the box in terms of the awareness raising that we set out to do. So some contact details here. Myself at IBioIC and Rachel at SINERGY. If you have any questions arising from today's session, want to find out about the support that we provide, or would like us to make some connections for you, then please do reach out. All that remains then really is for me to thank all of our speakers, and for those who were able to join us, and for those who put the session together, thank you very much, and enjoy the rest of your day.